Testing. Hello? 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 Oh, that's good. Back to it. 3 minutes, great. I think you can hear me now. Thank you for letting me know. You can let me know if you can hear me. I see the thing flashing. Ah, yeah. Blimmins. Well, there's no real topic for today. So the only thing I can hear is share some microbe slides. Yeah, that's about all I can think of, unless you've got some topic. I'll have to put in the chat 3 minutes. It's 3 minutes 20 it starts. If I can remember that. Right there. 3 minutes 20 audio starts. Sorry. There you go, that's for later. And I can post that in the description below. Thanks for letting me know, mate. I thought it was me. I thought it was me. Oh, yeah, okay. Yes. So if you've got anything to say, I'll give you 30 seconds because it's got a delay in about today's show. Otherwise, I'm going to start sharing some microbe slides on the rhizosphere from my class, Environmental, Dairy and Food Microbiology. It's one of the ones I'm doing this semester because I've got to share something. I've got to keep the stream going. And if you know, I had a few short shows lately, I don't like that. So, all right. I'm just going to start sharing. Because not much is being said. It's already 5 minutes into it. Slides. I'm joking. It's the wrong button. Share screen. We'll try again. So this is what I'm going to share. This is my course so far. That's the name of the course. So this is, I'll just go through a few of these, I think, because I thought these were pretty cool. This is from a different university. They don't really have much, what do you call it? Information on their slides. You'll see. It's different. Right, I'm ready to share. Is it opening? I think it is. It's trying. Oh, it did. There we go. How do I make it full screen? Let's go full screen. Thank you. It's not going full screen. Oh, dead set. This isn't really working for me at the moment. So it just wants to stay down low. Yeah, see this I can't remember which uni this is from. It says up the top. Xavier's College Kolkata. Okay. Different types of soils. So it's really, you can see the slides on the left there. Why isn't it full size? That's not the one. It's down there. It didn't work. Anyway, I'll just try and get on with it. Look through something interesting. Look how basic it is. This isn't the place where I usually study. How is soil made? Well, it takes over 1 million years to get 1 teaspoon of soil produced. And it's mainly from weathering. You can see all the different effects. And then you get the chemolithotrophic bacteria, which are the ones that work on the rocks. So it's another way. So like frost can also do it here, but it's another way to split the rocks. And it goes down and down. Functions of soil. Okay. Plant see how some of it's real basic and some of it's real extremely detailed. It's weird. This is from the conservatory, CEC it's called. This is the main body. And yes, soil particles is classified on size. Gravel, sand, silt and clay, and it's only to do with its size. Physical properties of it, of soil. Yeah. Clay, sandy, and sandy loam. I got a lot of sandy loam. It's horrible. It's hard to work with. It needs aeration, as you can see. Lots of organic matter need to be put in, and then it will help a lot. It'll turn into, like this, water will flow through a lot more cleanly. And here, no water flows through. It's bound up. This is very basic. Organic matter, clay, types of soil. No. It's pretty interesting when you're looking for different types of clay in the field. You can get, because there's a 1:1 clay, which is not good, and you want the 2:1 clay, which is produces isomorphic substitution, it's called, where the particles will exchange and then you'll get more attractions of potassium ions onto it. So the identification helps in that. Because you're going, what's all this? Why do you want that? Well, that's a reason how I've used it. Actually, I just put out a video on soil profiling on AussieCC website, on AussieCC YouTube, about all of this. Why is it important? Why do you want to know this? It's so blimmin' important to know what's going on underneath the ground. Because you look above the top of the ground and it is a general indication of what's happening underneath. So if things are really not going well, you'll know things underneath aren't going well. And the type of species too, because pH runs differently on different species. So by pulling it apart, you'll know how it's operating and functioning and how the nutrients are going to cycle for you. So it's really, really important in my eyes to get down at least a couple of horizons. So the O is organic, and the A, and then it goes, the B said no, there's another couple in between that. And the R, is the bedrock, the regolith layer. All the gold is. Component soil. Yeah, see, the A, E, B, E, yeah. So there's just little ones that mix in with it. And I'm not going to go through all them because I don't want to bore you. Different components of soil. That's what it's supposed to be, for it to be functioning properly and nutrients cycling well. You want about that ratio. So minerals, 45, and all particulate, and then you've got bits of other organisms. Look, humus, 1%, organic matter, 5, that's very rich, probably 2-3%, and it's still extremely good. Air and water is half of it. It's just a good way. So in other words, a lot of aeration. So if you're making up a substrate, you put the 45 into the 25, and that's nearly, a good ratio of it. So you can see how important it is. That's where the roots get their oxygen from. Soil minerals, okay. Cation exchange capacity of soils is extremely important if you want nutrients to cycle, because soil has a slight negative charge, so the positives get attached to it, the positive cations, and then there's a cation bonding strength, which is, that's a different slide. No, I'm not going to go into that because I'll have to look for that one. And why else is it? Well, when these elements get used up, they get replaced with a hydrogen ion, and then that gets exchanged with another ion, and that's how the nutrients cycle. Because the plants want different ions. It might be looking for magnesium here. This is going to get used up and cycled in through the roots and through the rhizophagy cycle. And then it's going to be replaced. So what's going to replace it? Hopefully not aluminium. That's the highest. That'll bind it up. Because see, that'll sit there and plants won't use it. They'll struggle. Sit there, nothing will do it. Aluminium's hard. It's not a very good addition to soil. It's the highest on the bonding strength table. Sources of minerals, fertilizers, biofertilizers, I hope. Well, this is just showing how they're all coming in. So this is just everything. Inputs from everywhere. Fuels, gases, spills, aluminium from cars, vehicles, all sorts of bad things can come into the soil. Attachments and non-polar. That has to do with the negative and positive charge, which we were talking about before with the cation and anion exchange, which is called the ion exchange. That's why it's very important. Attachments and microbes, here we go. So the clay particle is negatively charged. The bacteria has a slight negative charge as well. And then the divalent cations, which is the ones that can take it up, which attach to it and hold it because they're divalent. That means they can hold extra ions on with them, so they'll bind them up. So the attachments of bacteria through the divalent cation binding. That's how that happens. Another reason why the cations are important. See if there's any questions. No. This is the last one anyway. Soil pH is another thing that's extremely important. Without that, soil's not going to function. If it goes down one less unit, so if it's say 7 and it's normally down to 6, that's 10 times a shift in particles. So the one number is a 10 times fold. And then to go down again from neutral to 5 is 100 times fold. So it's very important you get in the right pHs. Oh, Jesus, summarizes too much. The soil water, this is like a speech in itself. Soil water gets in 3 ways: gravitational, capillary, and hygroscopic. The way when it percolates down through the soil particles, you want it to go down enough to support the plant and not run off and leach and pull nutrients out of it, but you want it to retain nutrients and hold in microbes and all sorts of other things. It's hard to summarize the whole thing. Wilting point is permanent wilting point. It's called when you'll see the plant start to wilt. That's when you know that the roots are starting to dry up and it needs water. It's a good spot to learn. Actually, I've got a video coming out on that. I've got a plant that I use for indicator plant for wilting because it uses more water than my garlic. So it's a good indicator plant. Yeah. And field capacity is the opposite end when you've got it and it's overflowing. So the water drains right through over the pots, out through the surface when there's just too much water. And you've got to be careful. You can't just go and tip it on and there you go, it's done, because it will go across the water top and not down through it. So you've got to make sure it drains in and it doesn't leach out. It's the hard bit. Soil aeration is important too for the microbes because they produce carbon dioxide when they respire. And if there's no pockets for the air gases to exchange or to release from, they'll build up and be in dangerous amounts. So it's one good way of measuring your respiration of your your respiration of your microbes through CO2. You can give a rough indication on how active your substrates are. So you can put a few shovels in of the O layer and into a bag with a CO2 meter and just watch it rise, and hopefully it'll just give you some indication on how the microbes, because once they're going through their cycling, it'll only last probably 20 minutes, because they'll be shifting from their different environment, because the moisture will change, the gases will change and all that sort of stuff, so they won't be functioning as well. But it's just a way that you can bind stuff up, or put a pot. If you've got a pot, even easier. Put it through a bag and meter on top. USDA classification of soil. Oh, yeah. Oh, that's the different visuals. If you want to know the different types, it's pretty. All right, where's the good one? What's the histosols? Vertisols, usols, monosols. No, I'm not going to talk about them, because none of them ring a good bell when he was going on about the soil science class. Thank you. Isn't that nice? Oh, it's Farmer Cheryl. Hello. Hang on, we've got something first. We've got genetic modifiers. Funny considering bauxite, the rock we get aluminium from, is a huge portion of the Earth's surface. It just requires a lot of electricity to separate. Yeah, mate, nothing to grow there. Or you could actually get specific. You could be good to work with that soil, because you would get stuff growing, and you could grab the genes out of it that would support the growth around aluminium, and put that in other plants. GMF says, I wonder if merely the ready presence of sulfur and its ability to readily bond could beat the aluminium to the bond. Now, sulfate is an anion, and the aluminium is the cation. So that couldn't happen chemically, I suppose. If that helps. I'm really bad at chemistry too, so you'd want to double check that answer. Farmer Cheryl says, are the slides available for viewing offline? I just turned in and missed a lot of the presentation. I show slides every week. I study at various universities around the world, mainly in India, and all of the slides I've got, I upload onto my playlists too. So there's many lectures there that you'll see on my playlists of various university courses, and all my classes, all my streams each week, they always show slides. I try to speak on slides, so it's for an easier explanation. And this is a bad example too, because this is a poor course. Like, these slides aren't the best. So if you like these slides, tune in on another show and you'll see some rad ones. Farmer Cheryl says, what is meant by soil sponge? I hear regenerative ag people talking about it. That's, well, you know, like your your sponge that you've got in your sink, how it absorbs stuff. It soaks up water. So if it's dry, you put it in, it soaks up, and it sponges and soaks stuff up. That's like the soil. So the soil's a sponge. It absorbs stuff if it's got its right cation and anions in place. Otherwise, if it's just like a desert, it might be real sandy and there's no organic matter for things to bind up on and things leach out. So that's an example of no sponge. So you can ask them about that one and stump them. I'm into busting myths, and I like showing things for stuff. So Farmer Cheryl, if you've heard anything like that, very difficult in the agricultural sector, please share it. I'd love to explain more about it and look at the different things. Science is a format where it's forever changing. So if someone's done something new, they could be true. But let's just see the science behind it and make sure it is true. Hope that helps. And drop some questions if it doesn't, Cheryl, but soil sponge, that's generally what it is. It absorbs everything. And it's hard to get stuff out of it. Sometimes things leach in. Their half-life is very long. Hydrocarbons, fuels, bad things like that, metals are really bad. So it just absorbs. GMF says, oh yeah, I think they're referring to the organic matter holding capacity, moisture holding capacity. Yes, that's well explained. GMF, every 1% matter, you get a 4% increase in holding capacity. Okay. Yeah. Huh. Interesting. GMF says, Cheryl, some of the post episodes are real diggers. Thanks for the nice words, mate. Yes, the organic matter is very important. The sponge. Well, the water, if they mean that by a source as a sponge, if they're talking about the water holding capacity, that's how I refer to it, as it's the water holding capacity. Yeah. I like all these different weird things. Thanks for that, Farmer Cheryl. I appreciate the ways that folk can help me here is by getting different people in and getting different talks so we can get different types of chat started up and interesting topics. So, I appreciate that. It's really good. So, should I continue then? See what else is? That was lecture 1. Or I'll do another one, I suppose. Why not? My voice is hurting a bit, so I'm just going to have a drink. And Cheryl, if you're still listening, I get into the more technical side of the ag, or I like to anyway. Keeps me interested. Thanks for the nice words, mate. Yes, more questions the better. I enjoy it. Righto, let's do it. Next lecture is share screen. Oh, rhizosphere organisms, interaction and function. Here we go. Look how basic it is. I'm sorry. Rhizosphere is underneath the soil around the roots. The rhizoplane touches the roots. The phyllosphere is above the roots. Adherents and rhizobium on legumes to form the root nodules. Nitrogen-fixing bacteria and form these little anaerobic shells where they produce nitrogen. Very cool. And this is vascular and vascular mycorrhizae network. See why that fungal systems work really well with soil roots. Some better than others. The reason is because see all the hyphae they put out, the mycelium grows and grows and grows underneath it. And then it's an extension of the roots from the plants. So it always stays moist. The fungus, it can transmit a lot faster than moisture, and it'll just really help a lot. And it also helps with phosphorus, most of them, and other stuff too. Rhizotonia infection, root bean, okay. So you want the roots to always look white. See how there's bits of black on it? That's an indication that something's going on. You'd know that from the top as well. You'd see that from the leaves would be twisty. They'd be doing something that's not normal. And then you'd look underneath, you go, oh, wow, look at this, I've got an infection. And then you'd have to diagnose it. And this would be diagnosed probably with the lab, I reckon. Method of overwintering rhizosphere fungus. Righto. So this is how fungus overwinters. So the fungal spores, they put out these little spores, and they will go and weather on the little droplets and stuff, and then they'll wait and they'll sit there, and the spores, you know, are very resistant. It's really only UVC and high temperature above 150 Celsius, what's that, 450 F or something massively high, 500 probably even, that affects them. So it just sits there and overwinters, and then it comes back to its right conditions again and germinates. Inhibition of rhizotonia by bacillus. So they've gone and put a bit of rhizotonia on a petri dish, and then they think, well, how are we going to fix this? I'm sick of it coming back every year. So they just use different species of inoculants, and this one was put in by bacillus. See, it only says bacillus. That's the genre. Doesn't even tell you what species. So it was put in by a microbe, and it found that it didn't grow. There was an area where it couldn't grow, so it worked. So this bacillus species is efficient against rhizotonia. So that's how you can test your if you've got persistent problems. This is a scanning electron microscope of bacillus inhibiting the mycelia. Isn't it pretty? So you can see the long threads. That's the mycelia, and the rice-shaped are the bacillus species. Induced systemic resistance and systemic acquired resistance in the plant body. So how you prime the genes into getting resistant to these types of infections in the future is you can induce them. So you get some type of microbe, for instance, or a methyl jasmonic acid, various ways you can injure them, all different types. I used to, you can use allelopathy, which is probably a pretty good one. And then you'll induce it, and it'll absorb it. And then it'll go in, and it'll start going systemic and go through the plant. And then you'll find it'll produce pathogenic resistant genes. So they'll be sitting there ready to go for when an invasion does happen. And the reason why that's good is because when a plant's under attack, it's usually under growth and development stage. And then it takes a while for it to activate its immune systems. So it stops its growth and development, goes into it, activates its immune, and then it'll work on it until it's fixed its sickness problem, and then it'll go back to growth and development. But if you prime the genes, its response is just so much faster. It'll basically see the problem and attack it straight away, and so it can still continue its growth and development. I've done it a few times, and it's like sort of fortifying your genetics. It's a good way for me to describe it. It's just a process of doing it. You can do it at home too. Stages of infestation of rhizobium in plants' roots. See, it goes back and forward, doesn't it? I'm not going to go through that actually. It just goes, the molecule growth, the spores there, it germinates, it bonds with the epidermis, and then it goes extracellular inside. It forms a root nodule. Different ways of nitrogen metabolization in rhizosphere. There's nitrogen-fixing bacteria, ammonium bacteria, like nitrosomus, nitrifying bacteria. So you'd have nitrobacter. And then you've got nitrate, which is one of the two nitrogen sources that the plant uptakes, the ammonium and the nitrate. You don't want denitrifying bacteria. So for those people who have some aquariums and they've got denitrifying bacteria in your aquarium to get rid of all the ammonia from your fish, that's not good because that's taking it out of the soil for where the plant wants it. So be careful if you do aquaponics. Mechanisms of phosphate solubilizing by rhizosphere bacteria. Aspergillium. Remember, Aspergillium brassiculaceae is better than Aspergillium lipiferium because it's produces indole acid as well. It's a Bacter, Pseudomonas, and Bacillus. Okay, very good. Fluorescent colonies. Yeah, Pseudomonas fluorescens. Isn't that cool? Pseudomonas fluorescens is a rad biofertilizer. Actually, really good for biofertilizer. I use it. Bacillus cereus is not good. It produces, what does it call that sickness? When you get sick in the stomach, come on, gastroenteritis. Streptomyces is a bacteria that they produce streptomycin from, and that's common in medical. You know, one of the antifungals. Pyrazine from Pseudomonas. Okay, actually it's Pseudomonas orificans. Why is that good again? Orificans. I'm into microbes for specific tasks to do one job for me. So if you know what it does and how it can work for you, you can apply it at the right time and get really good results. That's why I'm getting into the species name as well as the genre. What's this all about? I don't know them, so I can't talk about them. Plant growth promoting rhizospheric bacteria. They're good for doing all different types of things. Biocontrol, cytophore production, which is ion chelation, nutrient uptake, helps in nodulation, like legumes. Hormone production, which is all my microbes I use. I chose the ones that do a job for me and produce the phytohormones as well. And most of them do nitrogen fixation. I've got half a dozen nearly that do. I get free nitrogen pulled out of the blimmin' atmosphere and provided for free by my microbes that keep reproducing. What a bloody good system this is if you get into organics. Different activities of PGPRs. Get the badge table. Oh, no questions. Different PGPRs and what they do. So do some produce triglycerate phosphate or oxen or ACC dismutase? Okay. Cyclochrome, bacillus species. No, it's not specific enough for me. Down the bottom is, we've got all the Pseudomonas. I use Petita and Florens. I use Cochrocum. Mechanisms of pyridine. Oh, geez. Pythium, one of the Oomycetes, not very good. You'll see wilting on the plant. It'll wilt, even though it's not under a permanent wilting point from its water. Not good. Bacterial interactions in the rhizosphere. Well, good soil is supposed to have 50% pathogens, believe it or not. And because everything's cycling well in the immune system of plants functioning, everything's good. To summarize that, types of bacterial receptors indicating interacting with plant cells. Oh, they're pretty basic too. There's no real fancy looking syn ports there. Antiport, I won't get into too deep. Arena infection aids by insects. Arena is the thing that fungal, the bacterial infection that you see in carrots. There you go. So you pull your carrot out and it looks a bit spack, and that's because of that. It could also get into potatoes as well. You pull it like a black heart in the middle, produces. Oh yeah. Disease cycle of clubroot. Heteroconium, don't know that one. That's it. Trichoderma and Pythium. Wow. So trichoderma is really, really beneficial. I use Trichoderma hasarium. It's the only version of trichoderma that doesn't have indole acid as a one of its byproducts too, secondary metabolites that it puts out. So you can see here trichoderma is the long-looking line thing. It's the fungal hyphae. See it? With the thread. That's and then the pythium's all the dots in the middle. So what it does is the trichoderma is gone and grown through. It's an endophyte, so it's grown within the cells in the plant, and you can see it's isolated this bit of pythium. So it's pretty cool. So it's the interaction between two fungi. So one's extremely beneficial, and I'd suggest you get it and use it. And the other one is an Oomycetes, and yuck. Interaction between trichoderma and pathogenic mice. This is the process and how it happens, and it goes in great detail. I'm not going to go into detail. Multiple levels of rhizosphere interactions. So the different levels, you can see the plants up the top here, and there's a rhizosphere down the bottom, and there's all sorts of action going on because there's thousands of species of microbes around. The more the better. I say that's why compost is good, when the plant can decide what it wants. It's a sort of illustration of the rhizosphere again. There you go. That's weird, isn't it? Amoeba. Let's say what's the word starts with P, I think. Then they form all different weird shapes. Come on, what's that? It's not a bacteria, it's not a fungi, it's a no. Can't think of it, sorry. Oh, that's it. I should have asked actually seen these questions. No questions. 41. What do you reckon? Did you enjoy that? Because I'm ready for a break. And oh yeah, the next, I'll show you what is next week. So I don't want to do it this week. Phylosphere organises interactions. And then biofertilizer. I liked it because the biofertilizer was particularly good and they had quite a few names. And that's where I thought, sweet. I like the names. I like them to mention them so I can look up studies on them to see if they'll work for me and my environment. And also to see if they're symbiotic or not. Because a lot of microbes, they don't work with each other. Like humans. Try putting all of us in the same blooming room, look what happens. There won't be peace and laughter. So it's the same with the microbes. I try and see and just test them out, see what works really well. You should see what's the latest results that amazes me in the garden. Oh, my cauliflower I cooked yesterday was the size of a basketball. I'm not joking. It was a bit big. And I'm really not joking. You think, that can't be. It was. And my carrots, How many is watching? Do you want to have a look at my carrots? I'll give you a quick look. I was looking up. Actually, that's what I wanted to look up. World's biggest carrot. World's biggest carrot. Oh, you're joking. Are you joking? Is that all? I might have the world's biggest carrot. You're kidding. Oh, 10 kilo. Oh, damn. From these photos. And I'll show you what you're looking at. You go, he's full of it. Well, I'll even show you for real. That's big. But mine's bigger than that. Whoa, that's big too. It's way bigger than them. But yeah, there you go. Minnesota, US of mighty. What did you cook the size of a basketball? Oh, it was a, I'll go show you. I got one a bit smaller. It was a cauliflower, mate. Yeah, I'll give you a look. You want to have a look at the world's biggest carrot? Now, actually, it might be. How am I going to do this? So I make me video on and then remove that thing. Okay, I know how to do it. All right, so I got to walk with my computer outside. Let's go for a field trip. Oh yeah, you love cauliflower. Oh, you'd love this variety, mate. It's a different variety, and the variety has got. Hat on. But there's no snakes outside either. For them bloody things. The variety, it's, I don't know, it's new. You'll see. I don't know what it's called. I don't know names. But it's got all spikes and stuff on the top of it. And it looks all pretty, if that's a word for it. Righto. Okay, you can see. And the world's biggest carrot too, it's still growing. It's not the world's biggest carrot. I got a few of them because of my microbes. But here, I'll show you. Oh no, it's bolted. Here's one that's a similar size, but it's bolted a bit. There's my hand, if you can see that. So that's the cauliflower. There you go. Look at the prisms. See this? I've got no there. That's the one I ate. No, that's the one I ate yesterday. I cooked up. Look at the size of the stem. There's another one that's bolted. But yeah, really, that was the only good example. I think you can see it. It was all tight, and it looked like that. See those little, had heaps of those little things on top of it. That's going to flower. Everything's alive here. Little bees. It's humming. It's awesome. But anyway, these carrots. Here's my Brussels brocs. Puts out broccoli and Brussels sprouts. There was a carrot in there. Oh, dead set. It's dying. It is too. It's dying. No way. I haven't looked at it in so long. There it is there. There's one. Yes, hello. There's another big, this is the one I saw the other day. I thought that's pretty blooming big. And this is powering. Look at this thing. That's as green as. Look how big it is. That's that round. Yep. That's that round. That's one big carrot next to some very big garlic as well. Oh, there's another carrot there. Look at that parrot. Look how fluffy that is. Looks like a tree. Oh, this will be good. Oh, no, it's not that. It's got a split in it too, which is not good. I hope you can see this. Yes, hello. Hello, how are you? Oh, you're not missing out. He's just seeing something. Oh, it's on a small screen. Jesus. I really didn't know about that carrot. This is another one up here. I knew about that I've been watching. This one's grown. Oh, look at that. Isn't that full on? I love seeing expressions from. That's so crazy healthy. These are my Brussels sprouts. Look at the size of these things. Oh, this needs another spray. Look at that, mealybugs. Insecticidal soap and neem, 50%. Damn, let's have to do that today. Yes, I know. You're just waiting for something, aren't you? Good girls. Look at little legs. Yes, you do have little legs. And well, here's some more Brussels sprouts. These Brussels brocks. See on the side here. Look at that. So it's broccoli up the top, and on the side you've got these little Brussels sprouts. See, look at that. Oh, it's got bloody mealybugs too. Shit. God, you've got to be quick. Yes. Hopefully these will be all right. Yep, they're good and tight. Yes, hello, chooks. They give me 20 eggs a fortnight. I'm not kidding. I got too much. Oh, you want to see something? Oh, that doesn't really matter. But there's pineapples under there. What? There was snow the last few weeks? You can't be doing that. Yeah, well, I am. That's about it. For the interesting stuff, anyway, everything's just normal otherwise. Look at that, how healthy that's looking. Yum. I like it when it's got a thick, waxy layer on it. I just went and put some compost, you can see around it. Very, very healthy. Got a few varieties of silver beet. Some purple silver beet. Purple, heaps of antioxidants. I should show you the garlic field next time. Not this time. That's over there. It's looking extremely healthy. It's next year's garlic. All organic. It's very good. I've got to plant my corn soon. Get out and put in a few new rows of Brussels sprout. Look at this purple Brussels sprout. This is from seeds I tested from mine that I grew. Pretty rad, isn't it? I've never seen any purple Brussels sprouts. I don't want to eat them. No way, there's no mealybugs in it. Hey, is that got, that's, see, that's talking about, that's a resistance. So the test from this is really good. So the test from the ones over there is bad. I'll have to label those ones bad, and like this one is really good. What's this growing? It's a cauliflower, is it? It's white. It can't be a cauliflower. Looks like Brussels sprouts at the side, but that's a white thing at the top. Plant science, It's easy for you to do too. If you're interested, I can help you out. Ask plenty of questions. I can show you slides. Look at that. It's a purple. That's starting to bolt. That's finished its cycle. I did plant the seeds a bit late. See, that one's got some seeds up there, eggs, which I'm not happy with. Not happy. I'll squash them. They can be squashed. Now I'm happy. And the beneficial ones have like long tails on them. They're like lacewings, for instance. They'll be elongated a bit. So if you see some of those elongated ones, yeah, don't get rid of those ones. Look at that leaf in the middle. It's going to harvest some fungus. Perfect spot for stuff to breed. Oh, look at that sorrel. Yeah. But there you go. There's a little tour. Put on the spiders. Yeah, this I always had a problem with this phenotype for the mealybugs. So I got to get some some lacewings and Monterensis and biocontrol this. That's what my experiment is this time. So I'm not going to put any more neem and stuff on it. When I can afford it, I'll go and get some biocontrol and see if they can nibble it away. But keeping in mind too, biocontrol only works like right now. I should put it on because if it gets infested, it's not going to work because there's too many. They'll be out-competed. Have to wait till an event happens, weather event possibly or something, to reduce the numbers down. Oh, look at the goats disappearing. Oh, look at you. You want a treat, don't you? Righto, I'll give you a treat. Lucky I've got heaps of treats for them. One there, one there. A treat each. All right, easy as go. There you go. Good girls. Yum, yum. Rip into that. Is it gone? Oh, it's on the ground. You can see they love them. Right next to my spirulina. See my spirulina? Arthrospira and it's got a nice big biofilm on the top. They put out biofilm when they're stressed. So this went through a stress event, had snow on it too. This is 5 years old. See how it decomposes insects? So it puts them on the top. I don't feed this. This is organic. Spirulina. All gone. Yum, yum. What's the questions actually? Oh. Can I scroll up? I can see something about carrots. Oh, yep. Does vermiculite contain humic acid and fulvic acid? Does vermicompost contain humic acid? Yes. And fulvic acid? Yes. That can promote microbial populations and activate the soil. Yeah. Contains humin as well. You can tell by the colours. The humic substances has fulvic acid, humic acid, and humin in it. And the humin is lighter. Fulvic acid is a bit darker, sort of piss colour. And the humic is black, is really dark. So you can tell about that, so you can do your own diagnosis. Grilled, roasted, pickled. Styles, plastic under the Italian sandwich. Okay. If that didn't help, Cheryl, can you, when I answer things, just add to it? Just scroll down here for the yard looking outside. Looks delicious, even bolted in roast Really nice. Oh yeah, the collie. Oh yeah, that's what I like doing with my. Like, look at the broccoli. I've got to get rid of that. It's going to produce see those bugs? They're not good bugs. That's a white cabbage moth. See it flying around? Oof. Oh. That'll go now. It's gone. Anyway, bees humming around here, thank you. Soon it'll be a sea of bees. Anyway, getting back to, I got to read the question, sorry. Keep my phone too long. Are there any tips for growing carrots in clay soil? Yeah, add some aeration to it. So put some, ideally, organic matter. If you're talking about, most people, like you said, they don't have very good luck with the clay soil. I've got sandy loam, but if I had heaps of clay, I'd go and mix it with a lot of, look at that compost. There's chook manure, goat manure, cow manure is not very good, and turn it over. Oh, that was a carrot I just picked off. And because I was just thinking turning it all over. And turn it all over and do that a few times. That's what I've done in the field out there. So I've turned ungrowable land into growing land. You want to, because it retains too much moisture. So you want to make air pockets in it. So organic matter, sticks, twigs, will create air pockets underneath. Then bacteria can cycle through the air pockets and gases can exchange. At the moment, you're creating anaerobic pockets, which is so unhealthy for soil with the clay. Not you're creating, that's what the conditions in which your clay is at. So by aerating it, heaps of sticks, grass, just mow it, put it straight back in, let it absorb back in, go and put piles of this. Look at the pile, I got a truckload down the front, and I just wheelbarrow around, tip it out. I've done it 3 times this year, sorry. So every 3 months I'll put, make it black. Put this much layer around out there, make it black. It fills in all the spots, reduces weeds, adds nutrients, cycles really, really well. You have to make sure that the substance you get is free from contaminants and residues, glyphosate and problems that will actually cause in the soil. So you want to make sure it's sourced well, not just sourced from any person. I've made this, so it's, yeah, I know it's well. I've aerated it. I got rid of pythium. I had some Umiceti problems in it at the start, but I got rid of them, and keeps everyone healthy, doesn't it? And you can see the garlic, love it. Everything, look at the size of that garlic. Excellent. And the carrots. But yeah, to grow your good carrots, microbes is how I got them this big. And I'll tell you the secret, gibberellic acid. So if you don't use microbes, get gibberellic acid. Actually, no, I'm not going to even say that. Use microbes. There's a few that produce gibberellic acid. Actually, that Azosporium lithiferium is one of them. There's, I use, I think, 3 or 4 that produce. Look at this carrot. So this is from soil that's been, oh, it's dead. Oh, that's harvested. This is from soil that was too compacted. So I want to see, it's a purple carrot too, damn it. I want to flower it. Ugh. Doesn't look purple out of the ground, under the ground. There's a spin out. It's like 2 carrots. And you know that you don't get the Solanaceae disease that you get from the potatoes that have green spots, because it's only from the Solanaceae family, so carrots don't get it. So they can grow above the ground. Damn, I needed that yesterday. My meal. next fortnight. Yeah. So I just want to talk about carrots. Yeah, different condition for growing the carrots if it's too compacted ground. So I've done this, and I'm going to get my tiller out, and I'll be tilling this through all of these, making some new beds up. Cabbage white flies, moths. There's two over there. That's worthy of a thumb. That nice silver beet. That'll bounce back. Look at that was planted, I think, before, like 6 months ago. And suddenly I'm still in winter here. Massive stems. I blame that put that down with gibberellic acid. It's really good. Could need some pruning though. I think a bit thick. Look at all that stuff down there. That's just going to harbour problems. Can't look at it all. Oh, there's even a branch in there. That branch had no chance. Oh, you can't keep the one without the other. Look at her. Damn it. To break something else off. It's a pretty big thing. Small. You got to be fair to your pets, don't you? You can't. Here you go. Who wants this one? Okay, I'm going to go. That's the garden tour's done. There's some things for clay. Did I help the clay? GNS said some stuff. Right. Yeah, the humates is the byproduct of the breaking down of all of the worms and stuff like that, the black stuff you'll see at the bottom of it. Exactly what he said, GMF said. It's very helpful. So if you really want that, make yourself a worm bin, worm farm, out of some filing cabinets. Good looking plants, thanks mate. Until you're awesome, as usual. As usual. And I'll show you right now if you want to make let them go in. If you want to make a really good worm farm, I made one indoors, believe it or not, out of these. I had a 3-tier one, and so you get the totes put in plastic containers in the bottom. Bottom one will be a catch container. Second one will be your tote, and your next one up with your stuff in it. So the top two will leach into this one. This one will leach into the catch container. And the bottom one down here, you'll have a bowl with all the black stuff in it, with all your humic acids. With all your humic substances in it. So if you're really interested to make it, get more, you can make it that way. See one of the benefits too, with the spirulina. Let your pets decide what they want to drink. So sometimes I'll have a microbe brew. Sometimes I'll have spirulina, diluted, of course, because you can't have it too rich. It's very high in alkaline. It grows in pH 9 conditions, spirulina. And then sometimes you've got the normal 6.5 pH water, which that tests out of my faucet, my tap at 2. And that's hard to believe, and I've heard people say that before, but it's 2. And that's really low. All right. Done the tour. Go inside. Good. I like the tour, so I can explain stuff out there and why I do different processes and things like that, and how it's going to help me in my situation. because everybody's situation is so different and varies so much. We've got to try and work with what we have. And if you're like me with no money, you're going to even work harder. The organic thing's pretty labour intensive, that's for sure. GMF says, can't daikon and other radishes planted as a pre-crop do a good job of aerating and breaking up clay, so while also returning? Yes, you can use bioremediation, phytoremediation, if it's too heavily clay bound up. Excellent suggestion, GMF, if you're still listening to Farmer Cheryl. That's good. Really good suggestion. Check your pH first, Farmer Cheryl. That's probably the first thing you do with your clay soil, just to make sure that it's not too acidic. Because you might have to go and get some dolomite or some lime and raise it first. That's the only thing that you might have to do. And then you can go and put some crops in it that are root crops that support those types of busting ups of the clays, like GMF suggested. What's he said here? 2 pH tap. No, not pH. The east salts salinity, the ppm, parts per million of salts. Sorry. Geez, the pH out of my tap's 6.5. So I tested. I went around and did pH test. Actually, is that on my web? Yeah, I did that video a few weeks ago. I released that one. I got pH strips. So I tested my pond, my heaps of various things around there. My tank water, at the end of the tap water, to see if there was anything problems in the tap line. Yeah. So it was pretty. But no, thanks for clarifying that, mate. I didn't say it at the time, obviously. GMF says that is like straight citric acid pH. Like tooth and animal. We don't let sodas go below 4 because of tooth damage. That's pretty cool. Actually, the last week's lecture was all about citric acid. The professor there, he manufactures it at his plant, and he knows his stuff inside out. Next week's on acetic acid, Acetobacter. Oh, okay. Now I am less panicky. I've got pearly whites. I don't have any fillings. Can you still believe that? I went to the dentist a while ago, and he said, "You know why we don't like people like you?" I said, "Why?" He said, "Because we can't help you. There's nothing we can do. Your teeth just keep doing what you're doing." I went, "Oh, that's a good to know," because that was the first checkup I had in 20 years. And cool, it's good to know. So I just keep doing what I was doing, and yeah, everything's all right still. GMF says, I just made 38 jars of pickles. Oh, speaking of Acetobacter, holy mackerel. You feeding your village again? 38. That's a good effort. Acetobacter, yes. Yes, it's good fun making the microbes work for us. And I've just asked him actually a question, because he said that you might know this. He said that the reason why beer holds its gas longer is because of the proteins that have been used to produce the CO2. So it wasn't the CO2 added, it was the microbes that produced the Saccharomyces cerevisiae that actually produced the CO2, and it holds it longer through the proteins, breaking them down. So the question to him was, that hasn't been answered yet, that in my kombucha that I brew, how can I hold the CO2 longer in a bottle that's been opened. So I get 3 openings out of them, and on the third opening, on the third day, it's a lot gassy less. And I might burp once from drinking it. Well, usually I'll burp every mouthful because I like to have heaps of CO2 in mine. So that was a question that I've asked him, and I can't wait to see. Yeah. So I thought, can I add some sort of protein substrate at a certain stage that's going to allow this to happen better, allow the gas bubbles to hold longer, allow the, yeah. So that was a question that I had that I'd like to know, because it come up in his notes about when he was doing some fermentation, because the class is called, the course is Industrial Biotechnology. And yeah, it's pretty cool. I like it. I've learned a few things already. Holy mackerel, an hour. Talking about, yeah, time flies. Having fun. Well, that's about it. At least I got some, I think the microbes and the little, the garden tool was good. I've released some videos on I start to release maybe 1 a week now on my stream. It's not related to what it used to be related, but it's plant science, soil science, and microbiology related. So that's what I'm putting up now. See how it goes. So if you're interested, please check them out and leave your comments. And hope to see you next week again. We'll do this. I think we'll do the same thing again. Have some microbe stuff at the start, and then see how it pans out. And I like questions. Keep them coming. Thanks for the help again. You cool people in chat. It's, yeah, I learned stuff too with this shows. It's really good. GMF says, yes, naturally carbonated beer creates smaller bubbles. If you have 2 options, either chill the liquid and force in some more CO2 to dissolve. No, we usually use carb cap for a stand 21 bottle. He mentioned about separating the protein. Did you know about you can chill the beer? He chills the beer naturally from removing the large bubbles, protein, the large CO2 bubbles, and he only has small CO2 bubbles, and that keeps the beer colder when it's out longer, if that makes sense. But anyway, that's what he reckons that he does in India. The other method is to reprime. Yeah, I didn't want to do that one. I knew about that one. I didn't want to see what I could add afterwards, because that means if the bottle's open once, and then I'll put a teaspoon back in and then close it back up again. Hey, here's something cool that you might know. That I've got a species of Saccharomyces that activates in 3 degrees Celsius. What's that? 40F. And I think that's pretty rare, because I get carbonation inside of like a decent amount of carbonation in the fridge. So I'll only half fill my 1 litre bottles, and then I'll take some out, squeeze it back out of air, no air in it, put it back in the fridge, and within 24 hours, the whole thing's filled back up again. So it's Saccharomyces has, from what I understand, has used up some of the sugars and produced the CO2 as an electron donor, and made the bottle fill out again. So I thought that was pretty rad, because to get different strains of Saccharomyces they're available, but yeah, I didn't know if that's the one that commons in the beer industry, or if I've got a common one, or if I've got a rare one. That's because I've had the same one now for I'm using that same species for I don't know, a few years at least. Yeah, it's more plant science fun. Then leave it on the counter to warm up. Yeah, after tests to activate it. Yeah, well, there you go. I don't have to leave it on the counter, do I? Because of the species that I have, I've got a cold temperature strain for its growth and development. Ooh, I can share that too, because you'd produce how you reproduce it, and if you want to share it, your yeast cells is you produce it where it would go into spore form, and it's the spores that you dry out, and that's the bits that's regenerative. So that's how I could share my particular strain or have that strain tested. It's easier said than done, because it might be infested with other yeast cells and stuff from the air and things. You have to under aseptic, under microbe, under your laminar flow hood conditions. Here says germanium process correct, but there are a few other things that can do it as well. Ale yeast is genetically different from lager and wine yeast. Yes, I remember him saying that. Lager yeast specifically functions at those cold temperatures. Well, that means that's... Oh, shit. Oh, come on. I've been lucky to have like a photographic memory where if I close my eyes and picture things, I can bring up slides in my head that are actually written. I'm just trying to picture. There's a different microbe that they use. I only want to bring up that slide. I can miss. Jeez. Is it to do with ale? You use or is it the lactobacillus? The lactose. Exactly what you said. It's in last week's lecture too. I thought that was an interesting point. Something else, I'll read that. Also, if you're talking an Acetobacter, it has some bacteria as part of mixed colony that can function outside of ideal yeast temperature ranges. Oh yeah. I'm not sure about the Acetobacter. I just know the yeast bit, the Saccharomyces, the bit that produces the CO2. That bit, because I'm getting good CO2 production in the fridge, and that's not how it should work, from what I understand. Yes, we lacto ferment beer as well. Slightly higher temperatures make sour beers. That might be what it is. There's a different. All right, let's have a look here. You've got me blooming going now, even though the show's going for so. I'm going to pull up the slide. Industrial biotech. Short as last week. I'm going to share screen first so you see what I'm looking at. You'd find this course, GMF, fascinating. Because if your skill set, citric acid, is that? No, it might have been the previous week then. Look at the different. Let's write the two microbes. Found this pretty interesting too about Aspergillus niger, how it produces the black spores. So if you get any oranges from the fruit shop and let them sporulate up, and it's black, there's a high chance it's Aspergillus niger. Then you can go and use that to your advantage. Well, he uses it to his advantage and produces acetic acid. So citric acid. Solid, no. Nutrients required. No. Citric acid. I'm going to skip through. I'm sure it was liquid surface. Submerged ferments. Inoculation prep. No. CAM and stuff. No, that's right. This is all the different types of citric acid. You can get the mono on the can. No, I should maybe go on the last week's. Lactic acid. Oh, here it might be. History of labs. Well, I was checking labs first because it might be in here. Pediococcus. Actually, it might be here. Lactic acid pathway. The endomyelin pathway. It's going to be in the exam. Classification labs. 2 different types. This is maybe it. This is it, what I remembered, because it's the ambrin-myrin pathway, and then the heteros use the pentose phosphate pathway, which is the lactobacillus acidophilus, and then the homos is the bulgarius. That's the ones that he used, or that I found interesting because it was the casei as well, which, you know, lactobacillus casei. That's the Shirota strain derived from the Japanese professor over there. But yeah, that might have been it there. Spoilage reasons. Leuconostrop. He's just going at the moment. GMF's just going, oh, wow, I want to do this course. Because he's so familiar with all these blooming names. That's it there, I think. I'm happy with, yeah, because that was the 2 differences that I was thinking of, the homo and hetero in the lactobacillus fan. Because he goes on about the Bulgari, that's the one that he uses, because you've got to use for preservatives, and the lactobacillus is highly, highly competitive with its space. So it puts out the biofilm, therefore putting out antibiotics, which is a preservative. So it's very used a lot. And that was the one that he used. God, we got there in the end. Yeah, lacto. You feel like you're back in brewing school. Yeah, that's the way I felt. Being there, I haven't even done it. It was pretty fascinating, though. It's just interesting how we can use different microbes to do different things, and then we can go and derive different strains of the microbes to do different things, like the cold version Saccharomyces that I've got. It's rad. This plant science stuff, it's just fascinating. The world's so old, but we can do new things still. And we don't have to be in the lab. We don't have to have expensive machines or tech. We just got to have a little bit of education and our knowledge that we can share with our mates here, and we can have good results still. It's rad. I really like it. All right, this has been going on for ages, 1.5 hours. My voice was sore an hour ago. But how fun's it been? So I look forward to doing this again next week and talking more of the microbe stuff. That's what today's was on, wasn't it? I'll have to rechange it to microbes. We'll do more microbe stuff next week. That's the path that I think I'm going to tackle down the track to my plant science. I've done about as much as I can. I know how a plant works. I know how it operates. Blah, 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 with the cells and all sorts. You know how, then you got to just play with genetics. And then it comes to do microbes. What microbes can do for us and how they can influence plants, like my psychrophilic, and how I've kept pineapples in the snow to grow without heat. Well, you can't do that. Well, watch this show and you'll learn how to. We'll experiment together. GMF says, always a microbial good time, buddy. Yes, that's what I should really change it, something like that. Microbial fun, microbial goodness. Let microbes work for us. Yeah, I agree, mate. Thank you. This is also soil biology is a real game. Plants largely grow themselves. Yeah, and we can hybridize them. Woohoo! Like the Brussels Brocs. Good fun. Thanks, mate, for all your input too, GMF. It was good. Your scientific background helps answer questions too. So appreciate your time. And everybody else listening, all the people listening, thank you. Press the thumbs up if you thought this was cool. And I hope to interact with you again next week. Save all your questions till then. Let's have fun then. Have a lovely weekend. Thanks, FM Mushroom. Yeah, for mushroom. Cheers, CC. They say. Good on you, mate. See you two next week. All right, everybody. Thanks for listening. Happy breeding, happy growing. Good health to you all. Bye-bye. Thanks, buddy.